Hello everyone, welcome to today's live broadcast entitled LC Separations and Workflow Improvements for Environmental and Food Safety Analysis. I'm Jerry Workman, the Senior Technical Editor of LCGC, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by LCGC and sponsored by Advanced Materials Technology. Advanced Materials Technology is an innovative product development and manufacturing company focused on improving liquid chromatography separations. From pioneering fuse core technology in 2005, the Halo product line has changed previous conventional wisdom that high resolution requires high back pressure. Now in its 16th year, AMT continues to be a leader in novel, superficially porous particle development, enabling high efficiency and reliability for scientists involved in both small and large molecule separations, and delivers applicated solutions designed to meet specific industry challenges. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speakers. We are pleased to be joined today by Dr. Yelena Sopodnikova and Andrew Harron. Dr. Yelena Sopodnikova is a research scientist at the Agricultural Research Service, United States Department of Agriculture, USDA, in Windmore, Pennsylvania. Her research focuses on the development and evaluation of new, advantageous analytical methods for pesticides, veterinary drugs, environmental and emerging contaminants in food using gas and liquid chromatography with mass spectrometry. She has over 20 years of experience in the analysis of organic contaminants in food and environmental samples. Dr. Andrew Harron is an applications research scientist at Advanced Materials Technology, where his focus is on LC-MS applications. Prior to joining AMT, Andrew was the Thermo Fisher scientific and application scientist focused in HRAM and triple quad application and development. He also has experience in the government sector, having been with the U.S. Department of Agriculture as a research chemist in the Sustainable Co-Products and Biofuel Division, with a focus on LC-MS application development for co-products that are produced during the biofuel process. Dr. Harron received his PhD in chemistry with a focus in analytical and mass spectrometry under Dr. Charles N. McEwen at the University of the Sciences, developing and adapting new methods of ionization to both large and small molecule applications. In addition, he served as a faculty member in the Department of Chemistry at the University of the Sciences and as an adjunct professor at Villanova University. He is a member of the American Society of Mass Spectrometry, the American Chemical Society, and the American Oil Chemist Society, and is active in the Delaware Valley Mass Spec Discussion Group. Thank you both for joining us today. Yelena, please get us started.
Thank you for the introduction today, and thank you all for joining this webinar. Today, my presentation is on high throughput mega method for the analysis of diverse classes of contaminants, including pesticides, veterinary drugs and their metabolites, and environmental contaminants by liquid chromatography LC and gas chromatography GC coupled with tandem mass spectrometry MS. So first of all, why is it important to have advantageous analytical methods? Well, international trade of food and agricultural products is a huge market valued at about $4 trillion. And Each country during international food trade has regulations to protect human health, and those regulations include establishing tolerances or maximum residue limits for contaminants. And therefore, food analysis by different residue testing labs is also a huge market, exceeding $1 billion in cost worldwide. And all of this cost ultimately adds to consumers' cost of food. And when we talk about advantages analytical techniques that many labs worldwide seek, we need them to be, first of all, sensitive because of those tolerances or regulatory values. We have to make sure that those methods are sensitive enough to measure contaminants at or below tolerance levels. They have to be efficient and effective, relatively inexpensive, green, avoid toxic solvents, fast, so we can have fast turnaround and analyze as many samples as possible in shortest amount of time, providing information valuable to labs, consumers, and that is all leading to high throughput. So if we look into analytical process steps, the first step is when lab receives the sample is sample processing. The sample has to be homogenized. Then contaminants are extracted from samples. Unfortunately, during this extraction process, some unwanted matrix interferences are also getting into extract, which needs cleanup to avoid those interferences which might interfere the instrumental analysis. Here's the sample processing, and those are pictures from our lab. This is me holding a salmon, and the first process is to skin and fillet the fish, and the way we process samples or homogenize them is to use RoboCook with dry ice, which produces, as you can see here, very fine powder. Different labs use different homogenization techniques, and whichever technique your lab is using, the things to keep in mind that sample processing should always be a practical approach involving reasonable amount of time and labor. The analyst should keep behavior of analytes in mind, meaning degradation, volatilization, and the last but not the least is to avoid cross-contamination in between samples. For sample extraction, next step in our analytical process, many labs worldwide, including ours, using catchers. Catcher stands for quick, easy, cheap, effective, rugged, and safe. Here's the protocol or scheme for catchers sample preparation. We take X gram of homogenized sample, add internal standards, and add X milliliters of acetonitrile. Now, I want to draw your attention to the ratio of sample to solvent here. It is 1 gram to 1 milliliter. And after that, we either shake the sample and conduct salting out later, or we add, to stabilize the process, we add half X amount of gram of salts, magnesium sulfate and sodium chloride 4:1. Those tubes are shaken for 10 minutes on a vortex shaker and centrifuged for 3 minutes. After that, after filtration, it goes to analysis by LC-MS, and the remaining extract is subjected to cleanup. It can be dispersive solid phase extraction, DSP, or automated solid phase extraction, cleanup, and after cleanup, the samples are subjected to GC-MS analysis. By using this method, we can analyze several classes of contaminants from one sample listed here: pesticides, polychlorinated biphenyls, PCBs, polycyclic aromatic hydrocarbons, PHs, flame retardants, polybrominated ethers, which most of them been banned now. So other flame retardants are introduced, which were called novel alternative flame retardants. So overall, we can analyze over 300 contaminants from one sample using one extraction technique. And here's our, at the bottom of the slide, you can see our latest publication if you would like to take a look. Now, this all sounds good, right? But how about more polar contaminants, veterinary drugs, mycotoxins that also can be in samples because they are extracted and analyzed by separate methods? So the question is, can we also include them? Can we create a generic extraction method that would cover even more contaminants from one sample by one extraction technique, widening the scope of our analysis? And we were the first scientists thinking about it. Back in 2008, a research group from the Netherlands led by Hans Mol published a study in analytical chemistry where they attempted to develop a genetic extraction method. They tried different solvents and their combinations, and they developed a method which included acetonitrile water, 1% formic acid extraction with no cleanup to analyze for pesticides, natural toxins, and veterinary drugs by LC-MS. Quite recently, another research group from Denmark, also in collaboration with the research group from the Netherlands, published a study in food chemistry where they also attempted to get as many contaminants of different classes from one sample with one extraction technique. So they looked into pesticides, mycotoxins, and PAHs. And you can see a protocol they use. They use heptane, which is relatively non-polar solvent, acetonitrile, mid-polarity solvent, and water to extract a sample. And they had three fractions. One of them, heptane, on the top was solvent exchange to acetonitrile, taken through solid phase extraction procedure and subjected to GCMS. Acetonitrile fraction was split, cleaned by DSP for GCMS and injected into LCMS. And finally, aqueous fraction was injected into LCMS-MS2. So the goal of our study was to create a mega method, which we developed and validated, and we call it catcher-stir, which is more than catchers. We have developed and validated this method for over 350 analytes so far in fish, including tilapia and catfish, and also in animal tissues, including beef, lamb, and goat. And we published 2 papers listed here at the bottom of this slide for beef and catfish. We're currently working on a project validating mega method catcher-stir for over 500 analytes, which include different classes, pesticides, veterinary drugs, their metabolites, PCBs, PHs, PBDs, and other flame retardants, mycotoxins, and perfluorochemicals. And we hope to report on that project soon, so stay tuned. But for now, I want to show you how mega method, how catcher-stir works. So we take 2 grams of sample and we add internal standards, isotopically labeled internal standards. After that, we add 10 milliliter of 4:1 volume-to-volume acetonitrile water. So here I want to draw your attention to the ratio. In catchers, we had a ratio of 1 gram of sample to 1 milliliter of solvent. Here we have a ratio of 1 gram of sample to 5 milliliter of solvent, which creates some dilution. Now currently with high sensitive instruments, we can easily reach those tolerances, half tolerances, because the instruments are very sensitive, but this dilution provides less co-extractive materials in our extract. Second of all, you can see that compared to catchers, we use not just straight acetonitrile, but acetonitrile water combination, which helps to extract more polar analytes, such as veterinary drugs and mycotoxins. And then the procedure is very similar to catchers. We shake for 10 minutes, we centrifuge for 3 minutes, and then we take 200 microliter aliquot of this extract, evaporate it down with nitrogen, add LC aqueous phase, and ultracentrifuge. Now what ultracentrifuging doing here is helps to precipitate some co-extractive materials, and it works as a cleanup. It is also better than filtration because some studies have shown that when you use filters, then some analytes can absorb to filters and you can lose them or you can have lower recoveries. And then after ultracentrifugation, this portion goes to LC-MS-MS analysis. For the remaining extract, we decant it to tube containing 2 grams of magnesium sulfate sodium chloride. We shake and centrifuge, and then we take 1.5 milliliter of organic layer, and it goes to automated cleanup, conducted in parallel with GC-MS-MS, which I will show you on the next slide. So what is an automated cleanup? It is instrument top sample preparation, or ITSP for short, and here are 2 pictures. It is a robotic liquid handler that is installed on the top of our GC instrument. It uses, it also known as Micro-SP. It uses small cartridges pictured here on the top. They're 35 millimeters long and contain 45 milligrams of sorbents, including anhydrous magnesium sulfate, PSA, C18, and carbon X. 300 microliters of catcher extract is passed through this cartridge with a speed of 2 microliters per minute. And the cleaned extract is collected and injected into GC-MS-MS. So here's a scheme that shows you how GC run is conducted in parallel with cleanup. So we have cleanup step at 10 minutes, and then the sample is injected into GC, and robotic arm now starts the next sample cleanup. So our GC run is also 10 minutes because we use low pressure vacuum outlet GC, and this really saves time to conduct it in parallel, 10 minutes GC run and 10 minutes cleanup step. Here are chromatograms showing you on the top low pressure GC-MS-MS, and by the way, here at the bottom I listed our latest publication on low pressure GC if you're curious in fast GC-MS or GC-MS-MS. So chromatogram on the top shows you separation of pesticides and their metabolites and environmental contaminants in 10 minutes. And chromatogram on the bottom shows you that our UHPLC run is also fast. You can see that it also conducted in 10 minutes, and it covers pesticides, veterinary drugs, and their metabolites. Furthermore, we have 50 analytes that are both LC and GC amenable, and analyzing contaminants by 2 orthogonal techniques brings higher degree of confidence into their identification and measurement. So I was talking about cleanup, and what makes efficient cleanup is removing of co-extractives component and also achieving satisfactory validation parameters. And by satisfactory parameters, we call recoveries from 70 to 120% with RSDs below 20%. So to show you how efficient the cleanup is, here's a total ion chromatogram done by GC-MS, and in blue you can see chromatogram before automated ITSP cleanup, which shows fatty acids and cholesterol peaks. And in red, you can see chromatogram after cleanup. And by looking at the area peaks and doing calculations, we can see that 87% of matrix interferences are removed during the cleanup. And I'm sorry, I failed to say that was beef extract. Now let's look at lamb and goat extracts. So here's on the left, you can see a picture of 3 tubes. And the first tube, A, shows evaporated extract after catcher cell extraction for lambs. This is very complex matrix. B, tube B is after salting out, and tube C after ITSP cleanup. Now, not visual, but accurate representation of matrix, percent matrix interferences is presented here in this graph. And you can see from this graph that 99.97% of fatty matrix was removed by cleanup and salting out, providing efficient cleanup. So we established that cleanup is rather efficient. How about recoveries and RSDs? Well, when we validate the method, we follow USDA Food Safety Inspection Service protocol, which includes having reagent blink, reagent only in matrix match calibration curve, which allows us to calculate matrix effects. We also use 10 different blink muscles, and we have 4 levels of spiking, fortifying sample. One level is a tolerance level. So X here represents a tolerance level for each contaminant. So we validate at tolerance level, half tolerance level, 2 tolerance, and 1.5. For identification, we use retention time. When possible, for the majority of our analytes, we use 3 multiple reaction monitoring ion transitions per analyte. However, there are few analytes that do not produce strong enough stored MRM. In this case, we use 2, and ion ratios. So based on this validation, here are the results for catfish. This graph shows recoveries and RSDs for 106 veterinary drugs. And if you look into numbers on the top of green bar, they show how many analytes were successfully validated, meaning their recoveries were 70 to 120% and their RSDs were below 20%. So overall, 101 out of 106 veterinary drugs successfully passed the validation. If we look into recoveries and RSDs for pesticides, metabolites, and PCBs, the same graph, 235 out of 243 analytes successfully passed the validation, showing that this method works successfully for them. Another thing I want to point out, when we use this type of method validation, we have a lot of parameters to look at. And this generates around 200,000 data points that analyst has to look through. So in order to avoid exhaustion by looking through every single peak, we use summation integration for data processing. In this particular work, we used Agilent and SciEx software, and they both have summation integration function. For that, parameters are set up with peak start and end. Consistent retention time is important, and no human review. So, for example, for Agilent software, it looks like this. We set up parameters, and the software automatically draws a line from peak start to peak finish. And also I want to talk about matrix effects. I mentioned we're in reagent-only and matrix match calibration curves to look for matrix effects. Those are matrix effects in catfish, and usually we consider plus minus 20% matrix effects within the norm. And you can see it here in green box that most of our analytes, LC analytes, are shown in red diamond, and GC analytes show in blue squares. Most of our analytes are within the box, and of course we have some that are out and not behaving very well, because when we develop or when someone develops a method to have a very wide scope and to include as many analytes, diverse classes as possible, it's always some of them do not behave. So I want to summarize the advantages of our new mega method. First of all, we were reaching for wide scope to have as many contaminants of different classes of different polarities covered. So wide scope means that we can analyze more contaminants from one sample with one sample preparation, making this method efficient and effective. Cost efficient and high throughput. We can analyze more samples in short amount of time. And as I mentioned, our automated sample cleanup, ITSP, conducted in parallel with low pressure GC takes 10 minutes. And it also matched by UHPLC and SMS, which is 10 minutes each for run. With this, I would like to mention that all the trade names or commercial products I mentioned in this presentation were for the purpose of providing specific information for you viewers and does not imply any recommendation or endorsement by USDA. And I would like to acknowledge all of my colleagues and coworkers involved in this project: Steve Lehote, Alan Lightfield, Sergio Monteira, and Ederina Ninga were postdocs with the group working on this project. Savannah Simons, Robin Morton, and Lee Mayun helped with sample preparation. And with that, I would like to thank you for your attention, and I will be happy to take questions at the end. And now to you, Andrew.
Well, thank you very much for the opportunity to present here today. My name is Andrew Harron, and I'm going to talk a little bit about some of the work we're doing at Advanced Materials Technology. The title of my presentation is LC Separations and Workflow Improvements for Environmental and Food Safety Analysis. Okay, the first thing I'd like to talk about today would be Halo Fuse Core technology, and in particular, what makes our column special. Halo Fuse Core technology incorporates superficially porous particles, and this enables high efficiency separations, low back pressure, and high speed separations. And if we look at the Van Dienter plot on the lower right side of the screen, we can see a little bit of a demonstration of this, how at higher mobile phase velocity, we see our plate height is actually smaller. So we have a smaller plate height at a higher flow rate. And we can see how this can translate over here to examples of the SPP Halo C18 on the bottom, and on top here we have two examples of FPP column technology, which is the fully porous particles. So we look here at the top, we have two examples of FPP C18s. One is a 1.7 micron particle size and one is a 5 micron particle size, and we can see how these can compare to the Halo C18, which is a 2.7 micron particle size. So a brief comparison. If we look at the sub 2 micron particle for the FPP, we have 7,500 plates. And if we compare that to the Halo C18 with 2.7 microns, we see that we have 7,400 plates. So we have comparable plates. But the real trade-off here is the back pressure. You can see that the FPP column has 697 bar for the pressure, and where the Halo with the SPP technology has 339 bars. So we have much lower back pressure. So we have high performance, comparable plates, and half the back pressure. And we also have a faster analysis. Now conversely, when we compare this to the C18, the 5 micron particle, we would expect the lower back pressure with the larger particle size, which we see at 160 bar for the FPP as compared to 339 bar for the SPP. But we do see a real drop off in plates. So we only have 2,600 plates here for the 5 micron particle column, where we have 7,400 plates for the SPP particle for the Halo. And we can definitely tell that we have a much higher efficiency separation because we have essentially 3 times the plates that we have in the FPP column. And we also have faster analysis as well. So those are some of the benefits that can translate directly to your separation. Okay, which way? Now, column selection is paramount to a successful analysis, but sometimes C18 is not enough. And oftentimes this can be a little bit confusing and can be a little bit of a rather daunting task to figure out which column or which phase you should select for your separation. But at Halo, we have a lot of phases that can help you through this, and we have a little map here on the left side of the screen that can show basic trends and show the phases that we offer as well. So we can definitely help you resolve your problem. Okay, so now we're going to talk a little bit about these applications that we are doing currently, and we're going to talk about environmental and food safety applications. In the last 2 decades, the number of potentially carcinogenic compounds that has been reported in our food and our water supply has grown significantly. And the nature not only of these compounds, but also the matrices that they're in has become increasingly complex. We have seen from Dr. Yelena's presentation how recent advances in the Ketras-2 has significantly improved the sample prep methodology for complex matrices. And now we'd like to focus on the analysis, on the LCMS analysis specifically. And specifically, we're going to look at the analysis of pesticides, microtoxins, veterinary drugs and steroids, and how using fused core technology can enhance this analysis. The first application we'd like to look at is veterinary drugs and steroid hormones. These compounds are used in animal husbandry for therapeutic as well as growth purposes. The veterinary drugs are used for the therapeutic treatment of animals, specifically skin conditions, and there's a two-pronged reason for this. One of them is to keep the animal safe and keep the animal in a viable state to be harvested. And the second one is to ensure that no animal that has a disease will be able to transmit that to the human once the animal is harvested. So the two goals of it are to keep the animal healthy for harvestation and also to prevent any kind of animal disease which could affect not only the animal itself and the surrounding herd, but also humans as well. Now the steroids are used a little bit differently. The steroids are used for actual growth purposes. And in general, what happens is that animals are dosed with steroids for a period of time, and the function of the steroids is to increase the mass of the animal, to have the animal gain more mass and more efficiently for it to be able to transfer the feed that it consumes to actually packing on meat for the animal. And beef can be contaminated with large quantities of therapeutics as well as steroids. There's two problems with this. One with therapeutics, for example, if an animal is given a lot of antibiotics, it could start to develop bacteria which is resistant to antibiotics, and then this could transfer into humans once the meat is consumed. The second part with the steroids is that we're starting to see that, particularly in women, animals dosed with a high amount of steroids, you're starting to see that is transitioning into the development of cancer, specifically in women. And the steroid in question is progesterone. Now, progesterone is a naturally occurring steroid. However, in large quantities of it, we're seeing that we're starting to develop cancer in the breast tissue as well as the ovarian tissue. Specifically, those are the two cancers that are beginning to be linked to increased progesterone consumption in beef. So the column that we used for this was actually the C18 column, and the C18 is a workhorse column. It's probably the most famous phase or the most widely used phase. It is a universal phase for acids, bases, and neutral solvents. It has very good to excellent stability at low to mid mobile phase pH, and is a wide range of published literature applications. So it is a very widely used and well-used phase. All right, so our first example here is veterinary drugs. This sample was provided to us by the USDA. The USDA provided our sample here after extraction via the Catcher's 2 method, and they had spiked this sample with veterinary drugs. So we were able to detect 45 compounds, and we were able to classify them as well. And the concentration is 0.04 nanograms per milliliter, which is significant because we're in the low parts per billion range here. And as we heard a little bit about the MRL levels before, in the USA, the MRL level for a pesticide is low parts per million, potentially high parts per billion, depending on certain things. In Europe, it's actually 10 parts per billion. So we're able to detect these compounds at lower than the maximum limits for the MRLs in Europe and the United States. Okay, our next example is a mix of steroid hormones. This is an 8-compound panel. This is done in just over 7.5 minutes, and these steroids were clearly separated, and it's a very fast separation, and it's a very clean separation. These particular steroids were chosen because they commonly will arise in meat. You will see them sometimes. The primary one that we don't see is actually trenbolone acetate, TBA, We were not able to get that because that is a controlled substance and we don't have a DEA license, and that's a synthetic version of the testosterone. But we were able to detect testosterone, and we were able to detect these other compounds in just over 7.5 minutes. So what we wanted to do then is we wanted to look at actual real-world samples. So we went to the grocery store and we looked at organic beef and we looked at non-organic beef. These are two ground beef samples. One is organic and one is non-organic. And the compound that we found in there was progesterone, which you would expect because it is a naturally occurring hormone. It is naturally produced. However, if you look at the two on your left side of the screen, in the organic ground beef, we found it at 60 nanograms per mil, and in the non-organic beef, we found it at almost 7 times higher at 381 nanograms per milliliter. And then going a little bit back, as we've mentioned before, the exposure to the progesterone has led to the development of cancer, especially in females, because of this high amount of this excessive consumption, or this high amount. So we see an excessively high amount in the non-organic ground beef. So this is evidence that this particular animal was most likely dosed, because when they dose the animal with TBA, they also dose it with progesterone as well. So it's indicative that this much of a higher concentration, or this much of a higher value as compared to the organic, would be the use of some type of a synthetic being injected as well, as opposed to just naturally occurring. Okay, now we're going to talk about pesticides and environmental contaminants. And pesticides, most exposure is through the ingestion of contaminated foodstuffs. However, there's also potential exposure through contact, for example, Roundup or glyphosate. It's used on animal feed as well, although limited cases of contaminated meat or products from dairy have shown. So basically, it appears that there's limited evidence that suggests that the pesticides are actually breaking through and transitioning into the animal's meat as well. So an animal can eat this grain that has been sprayed with pesticides, and we don't see a lot of pesticides in actually the animal's meat. So it looks like it's not transitioning into that. So for this particular separation, we use the biphenyl. It's ideal for aromatic and pi compounds, or pi-pi compounds, or pi-pi bonded compounds, I should say. It has alternative selectivity to alkyl phases and is 100% aqueous compatible. And basically what the biphenyl phase does is it retains compounds through the same dispersive forces as the C18. However, it also allows for more polarizable substances to be retained. So basically these pi electrons that are available in the biphenyl phase create retention with the analytes, which are electron deficient. So if you have an electron deficient analyte, it's going to be retained. Because there are so many pi electrons in conjugation, you get much better retention for small and polar analytes. So that's the reason that we wanted to use the biphenyl for this. And if we see here, we have an example of 161 spiked pesticides and environmental contaminants that are detected at a concentration of 0.45 nanograms per milliliter in eggs. So we are lower again in terms of our 0.45 nanograms per mil. It's lower than 10 ppb for Europe. And all compounds were eluted in just under 20 minutes, and lower levels of detection than are stipulated by the EU and the US EPA. So we're seeing this. We're seeing this high sensitivity. We're getting all the compounds that were in there, and we're getting it in under 20 minutes, and we're getting it at lower concentrations than values that are stipulated by the EU and the US EPA. Okay, so now we're going to talk about microtoxins. And microtoxins are naturally occurring toxic secondary metabolites produced by fungi and have been linked to numerous toxic maladies in both humans and animals. And what's interesting about microtoxins is that they do show up in animals, they do show up in humans. So in the US, 70% of the grain that is produced is used for animal feed. So microtoxins grow on this grain, and they thrive on the grain. And so these animals eat this animal feed, which is infected with the microtoxins, and then the microtoxins survive that transition and they wind up in the animals. And what happens is that this can affect DNA, RNA, protein synthesis, and metabolic reactions in humans and animals. So not only do they survive the transition into the animal meat, they also survive the transition from the animal into the human as well. So they're very, very hardy. So unlike pesticides, where we don't see too much of a transition between the animals eating grain that has been sprayed with pesticides and the pesticides appearing in their meat, we do see instances of animals eating contaminated grain that is contaminated with microtoxins. We do see the microtoxins surviving that transition. For this separation, we use the PFP, and it is ideal for aromatics and electron-rich compounds. It has an alternative selectivity to alkyl phases, and it's useful in both reverse phase and in Hillick modes. And how do the C18 and the PFP compare? Well, they offer complementary reverse phase selectivity. So, for example, the C18 separates primarily based on the differences in the analyte hydrophobicities, whereas the PFP based on a complement of aromatic, hydrophobic, and polar selectivity. So there's three mechanisms of action that are operating in the PFP, which kind of gives you a lot of options for your separation. Okay, and here's an example of microtoxins that we were able to detect in beer. We have two examples. We have a home brewed beer on the right, and we have a craft beer on the left. And we were able to detect home brewed beer and four of the microtoxins in that, while 6 of the microtoxins in the craft beer. And so, as we said, these compounds or these microtoxins are on grain, so they survive the brewing process. So these beers is actually brewed with grain. So if you have infected grains and you brew those grains for your beer, you're going to see the microtoxins show up in the final product, which is what we see in these two examples here. Now we see more examples in the craft beer, which you would actually expect, and the reason is because craft beers often employ many different types of grains and non-traditional beer grains, which they would put into their brew, specifically to create new flavors. Is it too sweet? Is it too sour? They're trying to fine-tune their brew to appeal to certain craft tastes. So they access a lot of different grains that, again, are not traditionally used for brewing, so there's more possibility of contamination through microtoxin that way. But again, we can see that these compounds are very hardy. They do survive the brewing process. They do survive, from the transition from the plant to the animal to the human. So it's not too hard to understand that they would also survive the brewing process. Okay, now we're going to talk a little bit about emerging contaminants in food safety analysis, and specifically we're going to talk about PFAS. So PFAS, ordinarily when you think of PFAS, you think of the environmental impact, and it does have a drastic environmental impact. But we're also starting to see it transition into food, in particular, food that is inside of our food chain. So per and polyfluoroalkyl substances are chemicals have been used, of course, in a variety of things, from everything from carpet to firefighting foam to raincoats, pretty much the full spectrum of products you'll find PFAS in. And essentially what it is that their chemical structure includes both a hydrophobic fluorocarbon section and a hydrophilic carboxylate section. And that enables PFAS to be used for all these wide-ranging applications, but is also contributes to the negative effect it has, in that they're very stable molecules due to the hydrophobic nature of the fluorocarbon section. However, they are also highly reactive with polar molecules due to the hydrophilic nature of the carboxylate section. So they dissolve in water and they just sit there. So we just are constantly accumulating PFAS. PFAS is still being made today, and it is just consistently accumulating into our world. And essentially, we're starting to see the effect that it's having in the world in general. Specifically, we're starting to see it in food. The FDA just recently has raised concern about PFAS in food and milk, among some other things. And if we look this past year in North Carolina, unfortunately, a lot of these pets had very high levels of PFAS in their blood. And according to EPA method 537.1, we have a limit in our drinking water of 70 parts per trillion. And some of these poor animals had over 12,000 parts per trillion or 15,000 parts per trillion. So again, these animals are eating this, or these animals could be drinking the water, eating contaminated food, and the PFAS just accumulate. So our solution, we have the Halo PFAS solution. It is composed of two columns, the Halo PFAS delay column and Halo PFAS analytical column. And why do we need the Halo PFAS delay column? Well, specifically because PFAS are everywhere. They are ubiquitous. They're in almost everything. They're in Teflon. They're in your HPLC. And so it's very difficult to really try to analyze these compounds without the potential of having contamination. So that's why we have the delay column. We have essentially that column to catch the existing PFAS that are in the HPLC system, and they come from Teflon. They come from a lot of different parts in your HPLC system. So we use the delay column to just essentially delay the contamination from your instrument so we can actually get an actual good measurement on our sample, an unbiased measurement of our sample. And there's two different types of species. There's short chains and there's long chains. Around the year, in the early 2000s, long chains started to become less and less manufactured, but short chains have actually taken their place, and short chains are actually probably worse for you than long chains. So we have an example here of branched and linear PFAS isomers in spiked well water, and this is the comparison between the branched PFHXS and the linear version of it. And this is from a well water sample. So there's a well down here, not too far from us, and this was sampled from the water. There is 29 PFAS species in this contaminated water, and we can see that we can separate the linear and the branched, and we can also see that there's a higher abundance of the linear than are the branched, and that's kind of what you would expect because the branched isomers tend to mitigate down to the sediment, whereas the linear do not do that. So sampling, you would expect to see a little bit more of the linear than you would the branched, and that's basically what actually we see here. And this is a very quick analysis. This is done in just over 7 minutes. So how can fused core technology, how can it benefit PFAS separations? Well, in a word, speed. And you know, we're thinking now about the present, but in the future, things are going to get faster. High throughput's going to will be the king. You know, high throughput is where we're all going through, is what we're all going for, and it's going to continue to evolve that way. Mass spectrometers are going to get faster to get more scanning speed, and so people will want faster analysis times. So with this in mind, we designed a method for maximum speed to be able to separate all the compounds found in EPA 537.1, 533, and 8327. And we were able to do this separation in just under 5 minutes. We have all the 33 compounds ID'd. We have a nice separation of them. So we don't see a separation where it affects us. So any potential area where we couldn't ID them, we can. So it's not affected by, so the speed of the analysis has not affected the separation at all. We were able to provide adequate separation for the mass spec to be able to identify all these compounds identify the transitions, and we do have enough to be able to quantitate on that as well. So that's a very good separation. That's in just under 5 minutes. So in summary, challenging environmental and food safety samples require reliable and rugged LC column technology, and coupled with new sample preparations technologies such as the Catcher's Halo fused core columns play a critical role in new environmental food and safety analysis workflows, and that enables fast and reliable analysis of even the most challenging matrices. So Halo columns enable fast, efficient separations covering a full range of compounds for food safety testing, including veterinary drugs, steroids, and microtoxins, all the way to environmental contaminants such as pesticides, and an environmental and new emerging food contaminant, PFAS. So I'd like to make some acknowledgements. I'd like to thank Dr. Yelena for her presentation and for her work with us. I'd like to thank Dr. Lahote at the USDA as well. And I'd like to thank Dr. Charles Prowley, the Chief Scientist at STRIDE, Center for PFAS Solutions. And thank you very much. And with that, I'd like to turn it back over to the moderator. Thank you.
Thank you, Yelena and Andrew, for that informative presentation. Just a quick introduction here. Hi, I'm Amber Lowry, Senior Editor of Special Projects for LCGC, and I'll be moderating today's question and answer session. So before we get started on the Q&A, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And now on to our first question. Someone from our audience wants to know, why is ITSP not also used for LC with tandem mass spec sample cleanup?
Thank you for this question. It's actually a really good question. The first paper that was published on ITSP by Morris, I believe in 2015, was actually on pesticides analysis, and it introduced ITSP concept, and it was LCMS. The reason that we do not use ITSP cleanup, or any cleanup for that matter, in Catcher is because years ago, when we still used DSP, dispersive solid phase extraction, for cleanup, we noticed that many of LC amenable analytes would absorb on sorbents that we use in SP cleanup. And we did a study when we actually looked into absorption rate of analytes. So because LC is so robust, we decided to try with no cleanup. So back in Catcher's day, we just took aliquot of Catcher's extract and filtered it, diluted it, and injected it into LC to avoid this absorption and losing analytes through cleanup. So this is basically the same reason we do not use ITSP in LCMS in Catcher, because we don't want to use any analyte. So what we do now is we do ultracentrifugation, which helps to precipitate some matrix components, and then the sample is getting diluted and injected. And also, as I mentioned in Catcher, your extract is getting diluted during the extraction. Now, this is not to say that some people may not use any cleanup, ITSP or DSP, for LCMS analysis. It depends on the matrix, on analytes, and I guess on the preference of analysts, chemists. But in our opinion, cleanup is not needed for LC analysis.
Excellent. Thank you, Yelena. On to our next audience question. Can you summarize the main differences between quenchers and your mega method, and why the new method is better than quenchers?
Yes, thank you. I just mentioned some of that. Yes, so just like Catchers was developed and it basically took off, and the majority of labs worldwide use it because it's very efficient, cost effective, provides high throughput. We think that we believe and expect that Catchers also will take off and will be used in many testing labs. And the main differences, or I would say advantages of mega method catcher-serve versus catchers is, first of all, as I mentioned, we try to reach wide scope. So it's also important besides pesticides and environmental contaminants to include more polar contaminants, veterinary drugs, mycotoxins, in animal tissues, grains, and it is very convenient and to do it with one simple preparation from one method to get as many diverse analytes of different classes as possible. So this is one advantage. Another advantage is that, as I just mentioned in previous answer, because we use 1 gram of sample to 5 milliliter of solvent ratio, it gets diluted. So the sample equivalent that is smaller, and what gets injected is smaller. So that subsequently leads to less matrix components, matrix interferences, and better analysis. So this is another big advantage of catcher-serve compared to catchers.
Excellent answer, thank you. On to our next question. We've been hearing a lot about PFAS in food packaging. How concerned should we be about that?
Well, I mean, there is some in terms of how much of a concern it should be. There is some there. We should certainly have somewhat of a concern for it, because PFAS are in plastics, they're in food packaging, and essentially PFAS can leach into the food that we are about to eat. So there hasn't been a lot of information regarding what level of concentration in terms of the packaging or the plastic is too high to actually be used. So if we go back to basically the statute in terms of the EPA 537.1, that's actually 70 parts per trillion. The problem is that PFAS just tend to accumulate in your bloodstream, in your body. So it's very hard to get rid of PFAS. As a matter of fact, all of us as humans have between 3 and 4% of PFAS in our blood. So we have that much in us right now, and it's because we drink water, we eat food that has PFAS in it, and it just continues to actually just continues to be put into our systems, and we can't really get it out. So there is a level of concern. How much is a toxic level? Well, in terms of human studies, there's not a lot of information out there about what is the magic number in terms of how much PFAS can we tolerate before things start to happen into our bodies, like we start to develop cancers or things like that. But there are cases of actually animals essentially dropping dead from PFAS. So there is a lot of hot topics, and there's a lot of interest right now in that to figure out exactly what is the magic number in terms of a level for food contamination. Because what is the real concern is that we can't get rid of it. I mean, we just eat it. We just eat food that is contaminated with it, and it just continues to accumulate in our system. And that's the real problem with PFAS, is that it's just so hard to get rid of.
Thank you, Andrew. And, staying on the topic of PFAS, another member of our audience wants to know, but I believe you just elaborated on this, but they want to know, is there evidence of PFAS being toxic for humans, and at what concentration? So if you wanted to just add anything else to your answer previously, this would be a good time.
Yeah, sure. I mean, to go back on that, the real problem is that, we can't, it's very hard to get rid of PFAS in this, but they're starting to link some development of cancers to PFAS, especially if you look at, especially communities that live near high contamination spots. There is high levels that have been reported in people, and that has been linked to some certain types of cancer. So, but again, what is that magic number? What is the number that is very, very toxic to humans? It might be because of how difficult they are to get rid of. I mean, we could be at the point where a small level is just too much. I mean, we think about in terms of the water supply. In the U.S., 70 parts per trillion is actually the magic number. We can't drink water, or we're not supposed to drink water that has a higher concentration than that. So again, we're at that point where, you know, a very low level of PFAS can become a very high concern, not right now, but over 5 years, 10 years, you know, down the road, we're just constantly accumulating and growing our supply of PFAS that is actually inside of us. So it's hard to say what that level is. It hasn't been in food. I don't believe it's been reported what the actual number is, and part of the reason is because this is a relatively new field in terms of food to actually track PFAS. So, but it has the same properties as, you know, we would expect if we were constantly drinking water that's been contaminated with PFAS. We're just going to keep increasing our level. Same thing with food. If we're constantly eating food that has PFAS in it, we will constantly increase our level, which could potentially lead to some cancers.
Excellent. Andrew, thank you for tackling such a tough question, but unfortunately we're out of time for today. I'd like to thank the audience for attending and for participating in today's event. And I'd also like to thank our sponsor, Advanced Materials Technology, for making today's educational webcast possible. We would like to ask everyone in the audience to participate in a brief survey. You can see the survey to the right of your screen. You'll also receive an email alerting you when this webcast will be available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks again to all for joining, and we hope to see you next time. Goodbye.